Her research focuses on identifying cancer genetic risk factors, understanding how inter-individual and interpopulation genetic variations regulate the onset and progression in cancer, and developing strategies and tools to integrate genetics and genomics in the clinical management of cancer patients in Puerto Rico. She is the founder of the Puerto Rico Breast Cancer Genetics and Genomics Study, uncovering the genetic factors modulating the inherited breast cancer risk and tumor presentations in Puerto Rican women. Ongoing projects include the identification of genomic loci underlying and increased susceptibility to hormonal negative breast cancers associated with African ancestry and assessing the accuracy of breast cancer polygenic risk scores in populations of mixed ancestral origins. Es un honor para nosotros tener a la doctora hoy presentándonos esta conferencia magistral. Espero que le saquen mucho provecho. Con ustedes la doctora Julie Dutil.
Muchas gracias para la introducción y es un placer estar con ustedes hoy. Yo estoy bien encantada del tiempo que yo pasé esta mañana mirando las presentaciones y todo el trabajo bien interesante que están haciendo. Es que un placer estar aquí hoy. Voy a hablar en inglés porque si no nos vamos a tardar todo el día, pero en confianza, si quieren hacer preguntas en español, usualmente puedo entender. Es que no... y si quieren hacer preguntas, son los bienvenidos. All right. So, today I'm going to be talking about genomics and kind of give you a little bit of background of what has been going on in the world of human genetics and genomics, and really what we need to be doing in the future. So I'm really going to end the presentation with some of the new work that we're developing and that I'm quite excited about. And I'm going to talk especially about how it's important to have a lot of diversity in the genomic studies we're doing, and I'm going to give you the example of some of our work we've done here in breast cancer. So I have the official introduction. I'm going to also give you my genetic introduction. So this is where I'm from, a little town in the east of Quebec in Canada, but I've been living in Puerto Rico for 15 years. I spent actually more than 15 years teaching in first-year medicine in Ponce, and I recently moved to the Comprehensive Cancer Center to pursue my research there. And so if you look at my genes actually say a lot about where I'm from, because you will see that I'm mostly of French and German ancestry. Actually, my genes say that I'm coming from a small town where they make Bordeaux, which is a type of wine. And so your genes talk a lot about you, but I want you to keep in mind also that, while we're going to be talking about genetics, there's also the environment. So my children who have inherited half of their genetic material from me are being raised in Puerto Rico. So the other factors that are not genetic are also important. But today we're going to be really focusing on the genetic factors. So I have no disclosure. And so this is how we're going to structure the presentation. I'm going to talk a little bit about a big bias that have been seen in genomics research and really why it's important that we include diverse population in genomics. I'm going to give talk about some of our findings in the Puerto Rico breast cancer genetics and genomics studies, and then we're going to talk about some developing project, and who knows, maybe eventually you might want to get involved in some of those projects. So if we think of our genetics as humans, because I know that we've heard a little bit about the Drosophila megalogaster this morning, we've heard a little bit about the cow also. I learned many very interesting things. In terms of our human genetics, it's 3 billion base pair, and it's really, we've sequenced the human genome quite a long time ago, but we still don't understand everything. When I studied, it used to be our 20,000 protein coding genes, which is about 1% of our genome, and like everything else was called junk DNA. We don't call this junk DNA anymore. We now know that even outside of protein coding genes, there's very important regulatory regions that we also need to do a better job at understanding. And so our genes are is like an instruction book for our function, all of the functions of our bodies. And so when we talk about genetic diversity, also you will see that there's a lot of polemic and a lot of discussion in the world. And so we always have to keep in mind that even though we're talking about genetic differences, we're not talking about this to categorize people, but we're really talking about this and keeping in mind that we're much more similar than we're different, right? And so for every 1,000 base pair, there's only one base pair that would be different between two people. And so we need to keep that in mind. Also, we're looking at those differences because sometimes they can mean something in terms of disease risk. At the basis, we're more similar than what we're different, right? And so if we didn't have these differences, we would be looking like those monozygotic or identical twins here, right? And so these differences are the reason we all are a little bit different, have maybe different disease susceptibility. So sometimes small differences can make a big impact. If I see the big dog and the cat, right, thinking that this would be kind of the equivalent of our genetic codes, and you will recognize that these are 3-letter words, like our coding codon, right, for the proteins. Now, if I say the big dog and the cat, this is what you have in mind, right? If I only change one part of the sentence, it can turn into the big dog ate the cat, right? And this is not such a good story, right? So we can think of these differences as maybe little time bombs in our genomes or little differences that will make that certain people are more susceptible to develop certain diseases than others, right? And so if you think of your classic Mendelian genetics, is where we used to think of, you inherit what we used to call a mutation, which is like a very deleterious change in a gene, and that makes you at risk of a syndrome or a disease. But we know that this is not the whole picture. We also, and so these kind of Mendelian disease or these syndromes are pretty rare in the population. But we also know that there's some variance, and so now we call mutation more variance because it's got less negative connotation to it. And so we also have those differences or those variants that are a little bit more frequent, but they're not so impactful on risk. So they can increase moderately the risk of a certain disease. And then at the other end of the spectrum, we have these variants that are very common in the population, and then each of them have a very small impact of your risk of disease. But then if you happen to inherit a combination of these risk genes, then it can build your risk up. And so we need to understand these as well. Okay. Maybe if I can hold it, I don't know. Is that better? Yeah? Okay, good. All right. So we know that there, in terms of the kind of the more severe end of the spectrum, there are about 7,300 single gene Mendelian conditions. So if ever you want to do a career in genetics, there's lots of work. And we only know about the genetic cause for about half of them. And this is always increasing because we have better and better technology. Now, if we move to the end of the spectrum of these more common genetic variation and their risk of disease, this is what it looks like. This is what we know about which part of the genomes are controlling the risk of diseases. And so it's really hard to see because it's very small, but what you have here is your chromosome 1, and then all of these little dots are kind of genes or variants that have been identified to give a risk of disease. And this includes cardiovascular disease, metabolic disease, cancer. So we know a lot about genetic risk of disease. And so you could ask yourself, well, is there anything more to do? After all of this work, we've done hundreds of thousands of people with those studies to find the genetic risk. But in reality, we've always been looking at the same populations, and there's a bias in the people that we have included in the genetic studies. So this was the situation in 2009. It was 96% of the genetic studies were using people of exclusively European ancestry. And actually, when I got trained on analysis of this kind of data, we were taught to remove any outliers. So any people that look a little bit different than the European background, would just be removed from the analysis because this was a confounder. So fortunately, we've made some progress since that. And in 2016, we saw that this was an 80% European ancestry. But if we were looking at which studies were including non-European people, it was mostly studies out of Asia. And so China and Japan, when there's a lot of genetic research as well. And so if we look at our Hispanic or population of Latin America, this is the yellow part here, 0.54%. And so we've done very poorly at including this population in our genetic studies. Now, if we look in cancer especially, and breast cancer, because we're going to be focusing on that today, this is the last one here, you can see that we've done genetic analysis on more than 120,000 people, and then most of those, again, were of European ancestry. Now, if you're working in the lab with cellular models of cancer, so this is when you take what we call cell lines and to reproduce the disease in a laboratory setting, what we see, and this is some of our work where we're looking at the genetic ancestry of 1,000 cancer cell lines that are used commonly in research. And so what you see here, this little cluster, are people from different populations. So this is people from South Asia, you have people from East Asia, and this is the European people. And what we see is that the cell lines that we use in cancer research are mostly, again, of European origin. So even in the cellular model that we use in the lab, we lack the genetic diversity. And so if you're interested in the Hispanic cell lines, this would be the Puerto Rican cluster here. We have 3, all types of cancer mixed together. Colombia, Mexico, and Peru are here as well. So does it matter? Do we really need to do things differently? Well, if we're looking at what the US population will look like, it's a population that is more and more diverse with time, right? And so if we're looking at the younger generation, so people that are gonna have children now and are gonna be kind of taking over the demography in the future, it's more and more diverse. So we need to make sure that we know enough about the genetic of this population that we can really provide the same quality of prevention and tools and treatment for them. And so if we think of our history, we all are born out of Africa, and this has shaped what our genetics is today. And so as people moved out of Africa, there was exposure to different environments, to different disease, and this has created selection of certain variants or certain alleles in our gene, and that makes us today have different frequency of certain variant. And one of the first projects that was looking into this was the 1,000 Genome Project, and you can actually go online. This data is available. You can look at any variant you want, any gene you're interested in. And you're going to find this was basically sequencing of people from 26 populations around the world. And you will see that Puerto Rico was included. And so this is comparing genetic variation in these different areas of the world. And what you see is that the dark colors represent the proportion of all the genetic variants that are found uniquely in one population. So yes, most of our variants are shared either on the continent or around the world, but we have a certain number of variants that are unique to a certain population, including the population of Puerto Rico. We also know that, and so these variants that are unique to a population are those that we call private variants, right? And so we know also that the frequency of these private variants is lower. So these variants that are unique to certain population are more rare, but we also know that these more rare variants might be disease-causing. And so I'm going to give you one example where it really matters, and this is a study looking at variants in a gene called MYH9. And this gene predisposes to glomerulonephritis, which is a type of kidney disease, which is much more frequent in populations of African origin. And so what you see here is that the red is the variants or the combination of variants that cause the risk of kidney disease and what the frequency is around the world. And so if you look at the Africa, you can see that this risk variant is very frequent, But then as you move out of Africa, it gets less and less frequent. And so if you're a scientist and you want to study kidney disease, and all the people you have in your study are from European descent, it's going to be really hard to identify that variant because you're not looking where it is. And so this is, I think, and it's also not gonna explain why the disease is so much frequent in those populations. And so that's why when we think of this genomic study, we really need to go where the disease is more frequent, but also think of, making sure that we have the right people in the study. And so when I got here in Puerto Rico, maybe, 17 years ago now, I lost count. I wanted to do research, but I wanted to do research that would matter. I was coming from Montreal. I had done a postdoc in Japan, and I had studied the genetic risk of cardiovascular disease. And then when I got here in Puerto Rico, I wanted to do something that would really be connected with the community. So I wanted to have a study that would be advancing what we understand about genetics, but also be kind of useful for the population. And I found that in terms of breast cancer, there was really a need in breast cancer genetics. And we know that breast cancer is a very common type of cancer. I know that most of you have someone in the family or have heard of someone that have had a breast cancer. And we know that it's a disease that has a genetic component. If you look at sisters, first-degree female relatives, so that would be your sister, your daughter, or your mother, are 2 times more likely than the general population to develop breast cancer. So it means that if there's someone in your family that have breast cancer, independently of other environmental factors, you're more at risk of developing the breast cancer. And we know that these differences or this increased risk is because of genetics, not necessarily for other factors. And so what we wanted to do is that we wanted to understand how is the breast cancer genetics in Puerto Rico? Is it different than that of other populations? But also how can we use that information to improve breast cancer prevention and breast cancer care in Puerto Rico? So the study started in Ponce, which now looks like this. If you have been there recently, they have a whole new building for the med school. And this was a study where we were reaching out to the communities, but we also were reaching out to the physicians. And so we had surgeons, oncologists, primary practitioners that were referring patients for the study. And what we wanted to do is that we wanted to look at the whole spectrum. So we were looking for these rare variants that have a very strong impact on the disease risk, but also for these more common variants that have a minor impact on the disease risk, but that we could use for being able to understand better the risk of a woman to develop breast cancer. And what we want to do in genetics is that we need to think of the population as being a very diverse population, and within this population, we have people that are at very high risk of developing breast cancer. So can we identify these individuals that are at very high risk and maybe give them more surveillance, start the mammography earlier, and then also identify people that are at lower risk. Maybe we're doing too much screening for some people that don't really have a really high risk of breast cancer. And this was something that was really discussed in the last few years because Angelina Jolie, the American actress, actually was found to have a genetic variant that predisposes to breast cancer. And what she did is that she did a mastectomy, which means that she got her breast removed, even though she didn't have cancer. But this is something that people do when they have a very, very high risk of cancer. But so it was an opportunity not only to understand the genetic, but also to make sure that patients in Puerto Rico had access to the same genetic tests than people in the US, because 15 years ago, it was really, really not the case, I can say that. And so we started with those referral from oncology, surgery, family medicine, and community. And then we looked at what the family history was. And so if someone had a lot of people in their family with breast cancer, then we put them on the high-risk protocol. And then if someone had not so much family history, we would put them on the low-risk protocol. And so we had also controls. So we have women that had no history of breast cancer because we use that to compare to our cases so that we can see the difference, right? And so we've recruited so far over 2,000 women throughout that are from throughout Puerto Rico. So what did we do is that we took family history. So we sit down with every of these women, ask about if their mother had breast cancer, grandmother, cousins, and so on and so forth. We also took the pathology report because we know that breast cancer is a very heterogeneous disease, and so there's different subtypes of breast cancer, so we wanted to make sure that we had that information. We took questionnaire about all of the other non-genetic risk factors. For example, a history of pregnancy, history of breastfeeding. This is all associated with the risk of breast cancer. And then we took tumor samples, which is a part of the study I'm not going to talk about today, but we also are looking at the tumor genetics. And then we took blood for the inherited genetics, so the genetic variants that you got from your parents. And so this is the sample, what it looks like. And so we have mostly samples from Ponce, but now that I am in San Juan, we're going to resume the study and try to get a little bit more the north part of the island. We have also some work to do in the west. And so we see that most of our patients had invasive breast cancer and ductal. So this is a kind of the most common type of breast cancer. Now, what's important is that when you look at breast cancer, you can divide them into those subtypes. And one of the most aggressive subtype of breast cancer is what we call the triple negative breast cancer. So that's a breast cancer that has lost many receptors at the surface of the cell. And these receptors, we care about those because there's medication that you can give that will attack the cancer cell by binding these receptors. So if the cancer doesn't have the receptors, many of these treatment options that are working really well are not available. And they also have at the molecular level a more aggressive behavior. And we know that these cancer are associated, these what we call triple negative cancer are associated with African ancestry. And what we see here is that in Puerto Rican, that was 16% of our breast cancer, which is somewhat in between what we see for non-Hispanic Black and whites in the US. But when you look at Hispanic in the US, it's a 12%. And that's because Hispanic in the US is a mix. And we're going to talk about that. We have a mix of people from Latin America and people from Puerto Rico. And the genetic of this population is also very different. So we look at genes that were what we call our high-risk genes or highly penetrant genes. And interestingly, we find that almost everyone that had a mutation for this high-risk gene had the mutation, actually the same mutation or the same variant in the same gene, a gene called BRCA2 or BRCA2. And this was unexpected because usually there's a very diverse spectrum of mutation, and usually the BRCA1 gene is more common than the BRCA2 gene. And so when we looked at the genetic region around the BRCA2 gene, we saw that everyone that has this single mutation is the E13OX, so it's a mutation that is truncating, had a shared region around the gene. And this is what we call a haplotype. And basically this indicates that these people have inherited the mutation from the same ancestor. And this mutation was also located on a European chromosome. So this is something that was inherited from the European people that came to Puerto Rico. And so there's something in Puerto Rico which we call a founder effect. And so if you think of, if you look at the genetics of Puerto Rico, it's really a mix between European, African, and the Taínos, or Native American. And there's a founder effect, meaning that if you think of the people that were in Spain before coming to the Americas, there was a certain level of genetic diversity. But then not everyone from Spain came to Puerto Rico. Only a subset of people came to Puerto Rico. And what that does is that these people that came to Puerto Rico maybe did not represent all of the genetic diversity that was in Spain originally. And so this new population that was formed here is going to be less genetically diverse, but you may also have increase in frequency in some of the variants, or some variants that disappear, either or. It can go either way. Now, there's another thing that has impacted what we see in the genetic of Puerto Rico today, and this is what we call a bottleneck effect. So if you've had your basic genetics, maybe you've talked about this a little bit, and the bottleneck effect is when a population goes from a certain size and then it's drastically reduced to almost extinction. And this is what happened to the Taino, right? When the European came, it was really a disaster for the Taino population. And so the population size of this ancestor went down. And so we have examples of two in Puerto Rico. We have a syndrome called Hermansky-Pudlak, which is a very rare form of albinism, and it's maybe one person in the world out of 1 million had Hermansky-Pudlak syndrome. In Puerto Rico, there's 1 out of 25 that are carrier of the variant, and it's actually a high frequency in the area here in the northwest of the island. And then there's another syndrome called Steele syndrome, which is a collagen mutation, which is on an indigenous American bottleneck. So that means that there's some specific features in the population of Puerto Rico that you wouldn't know if you had not been looking at the genetics here. And this has important implication for those physicians that are treating these patients to be able to diagnose the disease properly. So one thing that we looked, in addition to these variants in the high-risk gene, we looked at genetic ancestry. And so when we think of the people in Puerto Rico, we think that it's a mix between African, European, and the Taino. And each person has a different proportion of that risk. And so the question we ask is, okay, if you inherit more African ancestry, does it changes your risk of cancer or the type of cancer that you will develop, and the other way around? And so this is looking at the ancestry of our study, the Puerto Rico Breast Cancer Genetic and Genomic Study. And you can see that it's really a gradient between the European cluster here, and this is the African Caribbean and African American in the US and African. And so you can see that there's some people that are almost 100% European. And there's some people that are as much African ancestry as a population from the Bahamas, for example. And so you really have a gradient. And in terms of the Native American or the Taino, this is this axis here, you can see that there's less variation, but there is some variation. On average, people have about 14% of their genome inherited from the Taino. And then in the mitochondrial genome, which is another part of our genome that we're not going to focus on today, but we have the other way around. So this is our maternal inheritance, and in this case, the Taino ancestry is much more prevalent. And if you think about that, that says a lot about the history of Puerto Rico, right? So men came to the island, and the Taino women were there, and that's why the maternal inheritance is more linked to the Taino than the nuclear genome. So when we look at, so we can use genetic markers. So what we do with these studies, that we have about 1 million genetic markers that are equally spread in the genome, and we use these markers to determine what's the ancestry of a person. And we know, so we use certain genetic variants that are known to be more prevalent in certain populations to distinguish what's the background of a particular person. And so what we see is that if you have a higher quartile or a higher proportion of African ancestry, you're more likely to have one of these aggressive triple-negative breast cancer. That's interesting, but can we use that to learn more about the triple-negative breast cancer? And so what we did is that we did an ancestry mapping study where we look at parts of the genome that would be enriched in African ancestry in individuals that have the hormonal negative receptor status. And so what you see here, this is what we call the Manhattan plot. So each point is one of those genetic markers, and this is the statistical p-value. And so what you're looking for is that you're looking for peaks that are sticking out of the background. This would be where you have an association between your genetic ancestry and a particular disease or a particular phenotype. So what you see here is that on chromosome 14, we have identified a region, and we're working now to do some functional studies where people that have an African ancestry are more likely to have the hormonal negative breast cancer. So we're hoping that in there, we can learn about the biology of what makes you at risk of having a hormonal negative receptor tumor. And that's also information that hopefully we can use to see if we can learn about new pathways or new way to target those tumors, right? And so we also have some association for these HER2 positive cases, which is another subtype of breast cancer that we're following up on. Now, in the world of breast cancer genetics, there has been one study that has been published by a collaborator in California where they show that at the chromosome 6, there's a strong protective factor. And this is the work by Laura Ferreman, who's now at UC Davis. And so there's a variant that when you inherit, you're protected against breast cancer. And it's a pretty strong effect. And this variant is exclusively coming from the Native American heritage. So you don't find it in people of European heritage or African. And so we looked whether this was important here in Puerto Rico, and really we saw no effect. And the reason for that is the Taino genome that we each carry in Puerto Rico is not very, very high, right? And so that's not, and what you see here, if you look at this graph, this is the proportion of Native American ancestry in people from Peru, which is the highest, people from Mexico and people from Colombia, and Puerto Rico is very little. And so in these populations from Peru and Mexico, this variant is very important to protect against breast cancer, but in Puerto Rico, really, there's not enough of the genome that is of Native American origin for it to really matter. And so what that means, and why I bring that point, is that when you do studies, and this is even true for the statistics in the US, they report Hispanic, Latinos, non-Hispanic white, non-Hispanic black, and all others, right? In reality, you're looking at very different populations. So what we see in Peru and Mexico is not necessarily true for Puerto Rico. And this is a screenshot from one of the figures from a consortium I'm going to talk a little bit about. But you can compare, see, for example, Argentina, where you have a very strong component of European, almost no African component, and then Puerto Rico is here where you have a much higher African component, and so it's the same for Brazil. So within all of these countries, or these populations that we classify as Hispanic Latino, we're actually looking at very different genetics. And this is just looking at the ancestry. It's not taking into account, for example, the certain variants, the founder effect, the bottleneck effect, which is likely to be different in this population. And so this heterogeneity has also impact. This is looking at survival for breast cancer by race and ethnicity, but putting into two categories, Hispanic white and Hispanic black, and you can see that there's a difference there again. And so we have to be very careful when we do these studies on making sure that we take into account that diversity as well. So we've talked about these high-risk variants that gives us high risk of breast cancer or other disease. But what we want to do in genetics, we also want to be able to use these variants that are less common to be able to predict the risk of a woman to develop breast cancer. And this is what we call the polygenic risk score. It sounds like a complicated thing, but it's really, all you do is that you add genetic risk variants at different sites in the genome. So for the breast cancer polygenic risk score, we have 313 variants that are in the most current model. And then the idea is looking at the combination of variants at these 313 sites in the genome and how they add up together to determine the risk, right? And so when we take this polygenic risk score that was developed in European population and we just try to apply it to Puerto Rico, what we see is that we have what we call flipped effect. And that means that in European population, one variant was identified as a risk variant, and here it behaves like a protective variant. And we kind of understand how this works in terms of the genetics, but it means that we cannot take the package of the polygenic risk score developed in the European population and then blindly apply it to any population, because we're going to have these flip effect, and these flip effects are going to be really important on being able to accurately determine the risk. And so when we're looking at the area under the curve, and I don't know if you've had statistics yet, but what we're looking at here is that the higher this number, the better a model is as predicting the disease risk. And so what we can see is that when we have, within the population of Puerto Rico, when we have people that have high African ancestry component, then the model performs less than in women that have higher European component. And so what we're trying to do now is to see how can we kind of tweak that model so that it works optimally for the population here. And maybe will mean that we don't include some of the variants, maybe will mean that we include new variants that are not important in other populations, right? And so what is next? And I'm gonna take the time we have left to talk about some of the projects that we have ongoing. And so, you know, when you do human genetics or population genetics or genetics in the field of medicine, it's really important, you need large number of people. So our study is 2,000 people. In genetics, it's considered like a small study. So the more people you have, the more you have the statistical power to be able to see those associations between genetics and disease risk. And so one very important aspect of doing research in genetics, and you know, as you're considering your future careers, is that you really need to have collaboration, and you really need to work with a lot of different other studies to be able to increase these numbers. And so our study has joined a consortium, which is called Confluence, and this is led by the National Cancer Institute, and this is pulling together all of the studies from all around the world and then doing the same genetic association studies. And so within Confluence, there's a consortium of all the Latin American studies. So we have up to now 32 different studies. So just like the study I just talked about, these 32 studies, we got together, we wrote a grant to NIH, which is likely gonna get funded, and this is gonna help us to continue to do the research, but taking 2,000 people from here and 2,000 from another study and all put this together. And this is actually the sample size that we have as of now, for which we have patients, cases, 25,000, and controls, 25,000, for which we have genetic data and then data about the kind of cancer they have, so that we can do that study, we can do these polygenic risk scores and continue that work. And so, and then we also want to make sure that we have a good database of what is the genetic in Puerto Rico. So the 1,000 Genome Project was actually my first research project in Puerto Rico back in the days, and I'm not going to say the date, I think that's the consensus here, because it makes us all very old. And this was done with Dr. Elinette Albino, who's at the recinto now. She was my PhD student at the time, and then Dr. Juan Carlos Martinez Cruzado and Dr. Taras Oleksik. And we were tasked in that consortium to recruit 70 trios, so mother, father, and children, and we had one summer to do it. And you know, we kind of got in the project on the late side, and we were, all the countries in the world were also getting their samples, and so we had very little time to find all of these families. And we had that plan where actually we had a lot of help from undergrad students in the colegio in Mayagüez, who were going to their hometown and distributing flyers to get people interested in participating to that 1,000 Genome Project, where we were aiming to sequence, in the end, there was 2,500 people sequenced, including about 200 from Puerto Rico. So this was great, but it's not a great baseline. It was the first time that we were getting data, and this is kind of the original paper, and it was the first time we were getting data about the genetic diversity in Puerto Rico, but 100, 200 people is still very little, especially if you're interested in looking at these rare variants that are likely to cause disease. And so now we're 15 years later, and actually Elinette and Elinette, who's now a professor, we actually are launching the next phase of this project. And in this time, instead of doing about 70 trios from Puerto Rico, we are going to be sequencing 100,000 people. And actually just saying the number gets me very nervous because we have a lot of work ahead of us, but that's going to be an amazing database because we're going to have So what we're planning to do is we're planning to do whole exome sequencing, which means that we're sequencing all of the coding parts of the genome. So it's only 1% of our genome. In certain people that have some syndromes, we might do whole genome as well. And the plan is that this data is going to be made available so that if students want to do some data mining, have a question about this data, we want this data to be shared. Kind of create a basic data set with whole exome from 100,000 people, which is kind of a dream. I think no NIH grant will fund that because the amount of cost is very high. But we partner with a company called Galatea Bio, and they are building biobanks of the Americas. So they're doing the same in Mexico, Colombia, and so we're going to start to get that, to bridge that gap in genomic data and get that data that we really need. And so we're going to be recruiting people in labs. We've partnered with the Laboratorio Borinquen, but eventually we might expand. And we're going to also get some clinical data, right? We want to associate the genetics with some of the phenotypes or some of the diseases. So we're going to get laboratory data. We're going to do survey, and then we're also going to access EHR, health insurance claims, and electronic health record. And so, so this is to be continued, but we hope that eventually any investigator in Puerto Rico that wants to do a genetic project can access that data, and actually also this is an opportunity to do more genetics research in Puerto Rico. And it's also an opportunity to discuss genetic in the community. So all the participants of the study are going to get a report on their ancestry, just like when you do one of these commercial tests, but in this case, it's going to be included in the study. And so you can learn about where your genes are from. And I think that, we think that using whole exome or whole genome data is the way to go to identify these genes for syndromes that are not understood at the moment. And we know that for families where there's a child with a syndrome, having a diagnosis can help in terms of, expectations, in terms of identifying like ideal management, and also thinking of new treatment. There's more and more clinical trials now with gene therapy, but you first need to know what's going on at the genetic level. And so I think I'm right on time, I'm gonna end with this. And so when we think of what we call precision medicine, which means that you use the genetic of a person or the genetic of a tumor to give a medicine that is appropriate, we can only do it if we are more inclusive in the people that we include in the study. And so I hope that with these upcoming projects, we'll be able to address some of these kind of gaps that we've had in the past in genomics. And so I would just want to mention all the people that work on the Puerto Rico breast cancer study. This is kind of the old team. We've transitioned now to the Centro Comprensivo de Cancer, but the study will continue. We're also setting up a genetic clinic for hereditary cancer, so that makes sure that people have access to genetic testing. And if you're interested to hear more about it, this is my contact information, so feel free to reach out.
Any questions? Pueden ser en español.
Los datos que usted presentó del breast cancer no me sorprende porque nosotros somos una población bastante homogénea. Pero hay la zona noroeste de Puerto Rico como nación es extremadamente homogénea, ¿verdad? Yo digo a mis estudiantes que le pregunten a sus abuelos que son primos hermanos o son tíos con sobrinas, ¿no? Así que hay un riesgo mayor. Ese riesgo lo vimos en el trabajo que usted presenta, pero ese riesgo de que tengamos una variante común en otro tipo de cáncer, ¿lo sabemos? En Puerto Rico, ¿puede haber otro tipo de cáncer que veamos esa misma variante?
Pues sí. No, está bien. Y se hizo, pues, no todos los cánceres tienen un componente genético alto. Se hizo estudio con cáncer de colon, la doctora Marcia Cruz, el síndrome de Lynch, y pienso que sí vieron lo mismo. Es que, y no es, lo pensamos por el BRCA2, que si eso era como regional a una parte de Puerto Rico, y cuando volvimos a los pacientes, se veía que venían de diferentes partes de la isla, donde esa homogeneidad va a tener un impacto grande es por los síndromes que son recessivos. Lo que pasa con BRCA es que, pues, no hizo la explicación, pero los genes de predisposición a cáncer son genes que se llaman dominantes. Es que se hereda el variante de mamá o de papá y solamente con una copia del variante, con el cambio genético, es suficiente para dar riesgo de cáncer. Y estamos hablando de riesgo alto, como con BRCA1 hasta 80%, que son 8 de cada 10 mujeres que tienen el BRCA1 que van a tener un cáncer de seno en la vida. Pero eso, una copia es suficiente. Con los síndromes recesivos hay que heredar ambas copias de papá y de mamá con un cambio genético. Y eso, como son cambios genéticos que son bien raros, es bien improbable que dos personas que no se conocen, que no tienen ancestría en común, de casualidad. Pero como, por ejemplo, Hermansky Pudlak es recesivo, pues cuando estamos hablando de personas que hay más consanguinidad en la familia, sí eso aumenta el riesgo, no tanto del variante, pero de ver el variante en forma donde ambas copias del gen en una misma persona está heredada. Y no es algo también porque se habla mucho cuando se da un curso de genética, ¿no? del asunto de casarse entre familia, pero muchas veces la gente no lo sabe. Eso es algo que es bien cultural también. Hay algunos países en el mundo donde está encouraged, ¿no? En South Asia, por ejemplo, hay varios países donde la tasa de pues, casarse con un primo es mucho más seguro que casarse con alguien que no se conoce, ¿no? Que son muchos de los aspectos que tienen que ver con la cultura también, pero en esas poblaciones se ven síndromes que no se ve en otros sitios del mundo por esa razón.
Yo tengo dos preguntas. La primera pregunta es, ¿en qué etapa está el proyecto de estudio de mutaciones en DNA mitocondrial? Y la segunda pregunta tiene que ver más con genotyping de los pacientes. Tenemos nosotros iniciativas aquí para conseguir fondos para hacer más genotyping de los pacientes de cáncer de mama.
Pues la primera pregunta sobre la de la mitocondria, lo que nos interesa en la mitocondria es que hay un fenómeno que se llama heteroplasmia. Es que pues tenemos mucha copia de nuestro genoma heredado en la mitocondria y lo que se ha visto es que los variantes que no son principal variante de la mitocondria, pero que son como escondidos en esa heteroplasmia, quizás están relacionados a riesgo de desarrollar cáncer. Pero el desafío con esto es que hay que hacer secuenciación como deep sequencing, que se llama, es que hacemos una secuenciación donde se lee la mitocondria como 1000 veces para poder ver esos variantes, que en la data que tenemos, es que estamos en este proceso de hacer la secuenciación, validar y siempre con la secuenciación de Next Generation, es una tecnología que hace muchos errores. Y eso pues se corrige en hacer más lectura, ¿no? Pero cuando estamos buscando por esos variantes de heteroplasmia que son más raros, siempre hay la pregunta, pues, ¿eso es un error de secuenciación o es un variante que de verdad es, pero no es común en la mezcla de la mitocondria? Eso está en una mezcla. La otra pregunta sobre genotipaje de los pacientes, pues los pacientes del estudio están... lo que hacemos es lo que se llama un array, un microarray, donde se puede hacer el genotipaje de una vez como más o menos, dependiendo del área, de 800,000 a 1,000,000 de marcadores de SNPs en una corrida. Es que eso lo hacemos sistemáticamente a todos nuestros pacientes del estudio, pero estamos haciendo también otros estudios para que tenemos la ancestría como... Lo que pienso es que todo estudio, aunque el tema no sea la genética, tiene que tener en consideración la heterogeneidad de la ancestría, es que estamos haciendo como de manera automática eso genotipaje en muchos de los estudios que tenemos en el centro de cáncer, pero también en Ponce. Ahora, hay fondo con cada de los estudios. El estudio de seno, especialmente, sabemos que eso va a seguir porque hicimos el consorcio, hizo una propuesta que se llama una propuesta R01 entre California, Puerto Rico y todos los estudios de Latinoamérica y no lo van a dar. Es que todavía no tenemos la carta, pero presentar one, es que si no la dan nadie tiene fondo. Es que vamos a tener fondo para seguir ese asunto de lo polygenic risk score y todo esto. Pero es un estudio colaborativo, es que está el liderato en California y en Puerto Rico. Puerto Rico va a manejar todos los estudios de Latinoamérica porque pues en el centro se habla español y pues por la comunicación es mejor. Y California va a manejar todos los estudios de Estados Unidos. Es que hay por lo menos por lo menos 10 investigadores involucrados en la propuesta, más gente como que son propuestas que son bien colaborativas, pero en genética eso es el mundo en el cual vivimos ahora. Hay que tener mucha gente, mucho fondo y mucha gente que pueden analizar data también, porque son data bastante grandes, que hay un componente de bioinformática bien grande.
Oyeron estudiantes.
El lab mío es mitad en el laboratorio y mitad en computadora solamente, porque hay que manejar esa data, eso es bien importante.